Welcome to our first CanVector National Thrombosis Seminar of 2026. My name is Joseph Shaw. I'm a hematologist and medicine specialist at the Ottawa Hospital, and I'll be your moderator for today's session. The title for is Biomarkers and Genetics for the Prediction of Venous Thromboembolism and Clinically Relevant Bleeding in Cancer Patients. So this is just a friendly reminder that this is a monthly seminar series that is hosted live throughout the academic year, featuring state-of-the-art thrombosis and VTE lectures presented by both Canadian and international experts. Recordings of the seminars are available after the sessions at the link that you see here. And just another friendly reminder that these national seminars are a self-approved group learning activity as defined by the Maintenance of Certification Program of the Royal College of Physicians and Surgeons of Canada. This is the National Seminars Planning Committee. I'm the committee chair, and you can see the other members of the planning committee here. So our speaker today for today's seminar is Dr. Danielle Roy. Danielle is a postdoctoral fellow at the Health Sciences North Research Institute. She completed her PhD in epidemiology at the University of Ottawa in the fall of 2025, where her research focused on risk prediction for cancer-associated VTE and bleeding. Her broader research interests include biomarker-based risk prediction, systematic reviews and meta-analysis, and predictive analytics using hospital data. The learning objectives are, as you see, listed here. So welcome, Dr. Roy. We're very happy to have you as a speaker today for a National Thrombosis Seminar. And just another reminder that after the presentation, there will be 5 to 10 minutes of Q&A. And if you guys have any questions, you can either include them in the Q&A chat box or get on your microphone and ask us directly. But I'll hand over to Dr. Roy.
Perfect. So I'm just going to share my screen. Oops. And share the presentation. Okay, just to confirm, you can see the slide?
Yes, yep, we can see
Okay,
it.
perfect. Okay, so hi, thank you very much for the invitation. I'm very excited to be here and presenting a bit on the research I did during my PhD. Like was mentioned, I'll be talking about my work on biomarkers, genetics, as well as machine learning for predicting VTE and bleeding in cancer patients. And I'll focus on the clinical motivation, the key findings that were observed during my thesis, and what this means for practice. And so I just want to start by framing the clinical problem. Why does VTE research in cancer matter? And so as most of you know, cancer patients have roughly a 9-fold higher risk of VTE compared to the general population. But most importantly, population-based data have shown that the incidence of cancer-associated thrombosis is rising, largely because patients are living longer and they're receiving more intensive systemic therapies, which has correlated with an overall better survival among cancer patients. And now the increase in VTE incidence among cancer patients is not good because it's one of the leading causes of death in cancer patients after cancer itself. And it specifically increases the risk of mortality by 3.4 to 6-fold. It can also lead to it can significantly impact a patient's quality of life. It can lead to delays or interruptions in cancer treatment. It can increase healthcare costs by 50 to 86%, and it can lead to higher chances of hospitalizations or outpatient visits. And so naturally, preventing VTE has become kind of more of a major priority in oncology. And so what has been done so far to help prevent VTE in cancer? So far, studies have evaluated VTE prevention strategies in 3 main settings, including surgically treated cancer patients, hospitalized cancer patients, and then ambulatory cancer patients receiving systemic therapy. Now, in surgically and hospitalized cancer patients, the guideline-directed thromboprophylaxis strategies are sort of more widely accepted. But when it comes to the ambulatory setting, practice is far more variable and not as widely accepted. And this gap is what motivated much of my PhD work. Now, specifically kind of zooming into the guidelines in ambulatory cancer patients, overall, the guidelines recommend thromboprophylaxis with apixaban, rivaroxaban, or low molecular weight heparin. And 3 of these guidelines specify the use of the Corona score to risk stratify patients, which is a validated point-based risk score based on cancer type, BMI, and some of your common blood counts, such as platelet count, hemoglobin, and white blood cells. However, despite the guideline recommendations, studies have shown that VTE risk assessment using the Corona score and thromboprophylaxis use as well in this patient population it remains underutilized in clinical practice. And key barriers potentially explaining this low adherence include the uncertainty of the net clinical benefit, the fear of anticoagulant bleeding, and then the modest predictive accuracy of the Corona score, as well as other existing risk scores. Now, I've included some statistics here which kind of highlight this dilemma more specifically. So some of the guidelines, as we just seen, they recommend thromboprophylaxis in ambulatory cancer patients with a Corona score of 2 or more. However, the cumulative incidence of VTE in this patients is about 9 to 10% compared to 5.5% in patients with a Corona score of 0 or 1. And now whether a difference in risk of 4 to 5% is high enough to justify thromboprophylaxis is debatable to some. And now the number needed to treat and the number needed to harm from the AVERT and the Cassini trials also highlight this dilemma because the data overall, based on the numbers, they suggest that for every 100 patients treated, about 4 VT events are prevented, but this comes at the cost of 2 major bleeding events, which reinforces the uncertainty around using prophylaxis in this patient population. And then lastly, the majority of the C statistics for the current VT risk scores in the external validation studies kind of lies between 0.5 to 0.7, which is not optimal. Typically, you want it to be much higher than that. And so this further highlights why perhaps it's underused in practice. Now, in contrast to VT, assessing a patient's bleeding risk is also an important aspect to consider when deciding on thromboprophylaxis. And there have been numerous bleeding risk scores, which are some of the key ones are summarized here, which have been developed to help with risk stratification. However, they have largely been derived in non-cancer population or have demonstrated unreliable predictive performance for bleeding in ambulatory cancer patients. And so this represents a significant evidence gap as effective risk stratification for both outcomes of thrombosis and bleeding is essential to guide safe and individualized prophylaxis in this high risk group. And so all in all, the main takeaway is that the current approaches are simply not good enough to identify which ambulatory cancer patients truly benefit from thromboprophylaxis, and that there is a need to better identify which of the ambulatory cancer patients will truly benefit from them without harming them. And so my PhD work was designed to address certain evidence gap to improve risk stratification in ambulatory cancer patients. And I focused on 4 main themes, which was directly supported or cited in existing research as gaps or areas needed to be studied. And so this include kind of genetic factors or inherited thrombophilias, so looking at their role in risk stratification, looking more into biomarkers, more specifically which ones might help predict VT events or bleeding events, which one are associated with events, and then also looking at bleeding risk more specifically. So it seems like most of the literature kind of focuses on VT risk, but not bleeding. And then the last theme was looking into machine learning and whether the use of a more complex methodology might help with better prediction as well. And so the first objectives was related to genetic factors, again, which include inherited thrombophilias. And what motivated us to look at inherited thrombophilia is that inherited thrombophilias have been established as a strong risk factor, or as strong risk factors for VTE in the general population. But whether they are also important risk factors in cancer patients on top of the cancer itself was not clear. Oops. Okay, so now as part of my thesis, I specifically conducted two different research projects related to inherited thrombophilias. So the first is titled Thrombophilia Gene Mutations Predict VTE in Ambulatory Cancer Patients Receiving Chemotherapy. And then the other one is a systematic review and meta-analysis on inherited thrombophilia and risk of VTE in cancer patients. Now, the first study was a post-hoc analysis of the AVERT trial, which for those not familiar with the trial, it was a randomized controlled trial comparing apixaban versus placebo for thromboprophylaxis in ambulatory cancer patients, which also had a corona score of 2 or more. And now during the trial, blood samples were collected at baseline and kind of throughout the trial. And we use the baseline blood samples to assess whether the patients had an inherited thrombophilia. And then we then performed an analysis looking at if there was an association between any of the inherited thrombophilias and with VTE or bleeding. Now, the second study was a systematic review and meta-analysis, which was focused on studies reporting an association between any inherited thrombophilia and VTE risks. But we did put a few exclusion criteria, which include leukemia or myeloproliferative neoplasms, and we excluded any tumor-specific genes as well. And so in the first study, we included all patients from the AVERT trial with available blood samples. And so overall, we had a sample size around 447. Out of those patients, there were 39 VT events and 39 clinically relevant bleeding events. And then in the systematic review and meta-analysis, we included 37 studies part of the review, in which 28 were part of the meta-analysis. And so in our two studies evaluating the risk of VT in cancer patients with specific inherited thrombophilias, there were 3 inherited thrombophilias which stood out as important risk factors. So specifically, we observed that the risk of VT was higher in patients with non-blood types, a factor V Leiden, or a prothrombin gene mutation, which kind of highlight that these may be important risk factors to consider. Now, on top of that, because the AVERT trial also looked at bleeding events, we also looked at the bleeding risk among these patients, and we found no increase in bleeding, which is reassuring because ultimately you want to kind of decrease the risk of VT without kind of increasing the risk of bleeding. Now, I won't go into too much details here, but this is just to say briefly show that, of course, with many systematic reviews, there were some heterogeneity in terms of patient population, study design, etc. So we performed quite a few different subgroup analyses to assess the robustness of the results. And overall, for the most part, the trend of increased VT risk in patients with non-blood types, a factor V Leiden mutation, or a prothrombin gene mutation was increased regardless of the treatment setting, the length of follow-up, the risk of bias, or if the genes were in Hardy-Weinberg equilibrium, which kind of further supports each potential true association between VT risk and these 3 inherited thrombophilias. And now, so we showed an increased risk of VT in patients with a non-blood type, factor V Leiden, and a prothrombin factor II gene mutation, which is great. But the next question was, can this risk be reduced with anticoagulation? Now, thankfully, because the AVERT trial included patients exposed to anticoagulation and patients not exposed to anticoagulation, it allowed us to estimate whether patients with factor V Leiden and non-blood types might benefit from thromboprophylaxis. So we performed this stratified analysis using incidence rates And as you can see from this bar plot here, we observed that patients with a factor V Leiden mutation or non-O blood types on anticoagulation, which is in red, had a much lower incidence compared to those who were not on anticoagulation, while the bleeding rates remain relatively more similar between the groups. And so what stands out here is the clear reduction in VT with using anticoagulation without a corresponding rise in bleeding, which reinforces the potential value of considering these two inherited thrombophilias when guiding prophylaxis decisions in ambulatory cancer patients. And so to conclude, the key findings from these two genetic studies are that ambulatory cancer patients with non-O blood types, factor V Leiden mutation, and the prothrombin gene mutations have an increased risk of VTE, but not bleeding, and that anticoagulation use can significantly decrease VTE risk without increasing risk of bleeding in patients with select inherited thrombophilias, making them potential candidates who would benefit most from prophylaxis. Now, of course, genetics is only part of the picture. So cancer is also a highly inflammatory and prothrombotic state. So we asked whether circulating biomarkers might capture hyper or hypocoagulability. For this theme, again, we conducted two different studies. So we first conducted a systematic review and a meta-analysis of all biomarkers studied for VT prediction in cancer. And then we conducted a study assessing the association between four inflammatory and cardiac biomarkers used in cardiovascular medicine and VT and bleeding, which was also done using data from the AVERT trial. And now, what was the rationale for performing a systematic review and meta-analysis on biomarkers for the prediction of cancer-associated thrombosis? Well, when I initially was kind of diving into the field and reading different studies in this field, I found that there were many, many articles assessing the relationship between biomarkers and VT in cancer patients. And it was almost in a way chaotic how many studies looked at the same associations. And so I wanted to bring order on what has been looked at, what perhaps should be looked at further, and what perhaps we should be paying more attention to, in hopes of potentially trying to move the field forward in a more, I guess, systematic and staged approach, rather than keep looking at the same associations over and over again. Oh, these are a couple more studies. And so the aim of the study was to summarize current evidence and identify biomarkers that commonly predict VT in cancer patients. In this review, we included all observational studies focusing on adult cancer patients, reporting an association between any blood biomarker which was measured at a specific time point, such as pre-chemotherapy or at cancer diagnosis or preoperatively. And it had to be in association with a first VT event, so not any recurring events. And so overall in this review, we included 113 studies, of which 50 was part of our meta-analysis. And the characteristics of the 50 studies which were included in the meta-analyses are summarized here. So we can see the majority of the studies were cohort studies with a slightly higher percentage being prospective. Then approximately 30% of the studies included various cancer types, whereas the remaining studies focused on a specific individual cancer type. Now, approximately 86% of the studies reported on overall VT as the outcome, with a few reporting on DVT or P only. And then lastly, the majority of the studies had a low to moderate risk of bias. And so according to our meta-analyses, there were 9 biomarkers which were identified as potential predictors, which differed based on timing of measurement, specifically at the time of cancer diagnosis. But markers such as higher factor VIII levels, lower time to peak thrombin, lower hemoglobin, or hemoglobin, sorry, below 100, and then white blood counts over 11, were markers that showed the strongest association with subsequent VT risks. And then pre-chemotherapy, elevated D-dimer, elevated fibrinogen, lower mean platelet volume, and then neutrophil lymphocyte ratio over 3 were associated with increased VT risk. And then lastly, higher preoperative platelet counts was an indicator of a higher VT risk in the surgical setting. And so to conclude this theme, so we comprehensively reviewed and this study, we comprehensively reviewed and meta-analyzed various biomarkers that were evaluated in the literature. And we identified 9 biomarkers which may be useful in the prediction of cancer-associated VTE. And the associations we observed can provide important insights for future research efforts aiming to improve VT prediction in cancer. And hopefully the findings that we observed will help move the field forward. And now moving on to the second biomarker study, which we looked at inflammatory and cardiac biomarkers. Now you might wonder why inflammatory and cardiac biomarkers. So we chose inflammatory biomarkers because cancer causes a pro-inflammatory state, and then chronic inflammation has been shown to be associated with an increased VT risk, sort of via a couple different mechanisms. So this is summarized in this image here according to Virchow's triad. So for instance, inflammation can cause hypercoagulability or even affect stasis through the release of pro-inflammatory cytokines, which can activate the coagulation pathways. And inflammation has also been found to cause endothelial dysfunction as well, which also is a contributor to that promotes a prothrombotic environment. Now, moving on to cardiac biomarkers, we looked at cardiac biomarkers, specifically HSCNT and NT-proBNP, because they have been associated with VTE in non-cancer populations, and they are often elevated in conditions that predispose patients to clotting or bleeding, such as heart failure or endothelial dysfunction. Now, on top of that, these biomarkers have already been used in other validated cardiovascular risk scores like the ABC bleeding score and the ABC stroke score in atrial fibrillation population. And so we wanted to explore whether they might also have value in the oncology population. And so in this study, again, we included data from the AVER trial. And during the AVER trial, the blood samples were collected at baseline, 1 month, and 3 months. And then using these blood samples, we analyzed the biomarkers GDF15, CRP, NT-proBNP, and HSCNT. And we assessed the relationship with VT and bleeding risk using fine and gray regression analysis. And from our adjusted regression analyses, our main findings were that elevated GDF15 and increasing levels of HSCNT from baseline to month 1 were associated with an increased VT risk, whereas elevated NT-proBNP and CRP at baseline were associated with a higher risk of bleeding. And so these biomarkers may help optimize risk assessment for VT and clinically relevant bleeding in ambulatory cancer patients starting chemotherapy, as these were the associations we observed during our study. Now, the study presents the hazard ratios for the 4 biomarkers we evaluated in relation to VT and bleeding risk. And this quantifies the relationship between the biomarkers and VTE and bleeding, but to better portray the predicted risk and facilitate external validation or sort of like replicate or help other studies replicate our findings, we also developed nomograms. And so these are the 4 nomograms that we developed. So there's 2 of them predicting VT and bleeding risk at baseline, and then 2 of them that are used that uses 1 month data. And again, we included them to allow other researchers the opportunity to validate or build upon our findings. And we also provide an explanation on how to use them in the article. But essentially, what a nomogram does is that it assigns each predictor points proportional to its relative contribution to the outcome. And these points are then summarized. So these are the points at the top. These points are then summarized in order to generate a total score which maps to a predicted probability value for an individual patient. And so in conclusion for this study, we showed that elevated baseline GDF-15 was associated with increased VT risk, while elevated baseline NT-proBNP and CRP levels were associated with a higher clinically relevant bleeding risk. And then lastly, changes in HSCT over time could also predict VT risks. And so overall, these biomarkers, again, they may help optimize risk assessment for VT and clinically relevant bleeding in ambulatory cancer patients. But of course, further large prospective studies are needed to confirm our findings and the utility of these biomarkers as well. And so now on top of thrombotic risk, bleeding risk is also an important consideration because while you can prevent VT using anticoagulation can be the opposite of beneficial for some patients by increasing the risk of bleeding. Therefore, both the risks of thrombosis versus bleeding should be weighed when deciding on thromboprophylaxis use. And that's why 2 of the 4 studies so far we concurrently looked at bleeding risk on top of VTE to kind of identify which of the risk factors for VTE might also be risk factors for bleeding. And this was done to hopefully inform future modeling studies. And so specifically, we in our thrombophilia gene mutation study using the average trial data, we found that none of the 8 inherited thrombophilias that we looked at were associated with bleeding risk. And then in our inflammatory and cardiac biomarker study, we found that CRP and NT-proBNP might have value for predicting bleeding events in cancer patients. And now on top of that, we also developed bleeding risk models, but it technically falls under the next theme of machine learning. So I'll present it as part of the next theme. And so last but not least, the last and final theme is machine learning. And as I mentioned earlier in this presentation, existing risk models for VTE specifically, they perform modestly. And so our goal here and the question we asked is, can more advanced modeling approaches do better? Can the use of machine learning methodology improve the prediction performance and help predict some of these complex outcomes? And so this led to the final study part of my thesis. The study is not yet published, but it's titled here on this slide. But the aims of the study were twofold: to determine if using more advanced modeling methods improves the prediction of VT and bleeding events in this context. And because we looked at various biomarkers and genetic factors in the average trial, the other aim was to determine if the addition of biomarkers and genetic factors that are not routinely available in clinical practice helped improve model performance. And so once again, we use data from the average trial, and we specifically developed 3 different models. So we developed a logistic regression model with L1 regularization, which is essentially simply a logistic regression model with automated variable selection. So instead of me specifically choosing which variables to include using backward or forward strategies to pick the variables, I instead included an algorithm which basically automatically did it for me. And then on top of that, we also developed 2 XGBoost models, one which was using all possible variables, and then another which only included routinely collected variables. And briefly, XGBoost stands for extreme gradient boosting, which is a more complex, advanced machine learning algorithm, which is often seen as a machine learning model which can achieve high predictive power. But it comes at the expense of interpretability compared to logistic regression models. So you can see this is a common graph you've probably seen across the internet. But the more advanced models you go, it might lead you to higher predictive power, but your model interpretability will go down compared to your more simple models. And now in the manuscript itself, we report other performance statistics, but for the sake of the seminar, I'll only focus on sort of the most common performance measures that's used in modeling, which is the area under the ROC curve. And overall, our results suggest that the XGBoost model, using all variables, including non-standard biomarkers and genetic variables, performed best for both outcomes. Although for clinically relevant bleeding, the XGBoost model, which only included routinely measured variables, was not far behind in terms of performance. And so overall, what this shows is that both using an advanced machine learning model on top of utilizing non-standard variables that capture potential other mechanisms of risk was helpful in improving prediction. And so since the advanced XGBoost models performed best, the next step was to understand why, specifically which variables the model relied on the most for its prediction. And a common way to make machine learning models more interpretable is by looking at a concept called variable importance. Variable importance in machine learning can be calculated in different ways, kind of using different statistics. But whichever method you use, it's simply a metric that quantifies how much each predictor contributes to a model's predictions. And so with that being said, in order to make our models more interpretable in our study, we looked at which variables were most important and helped with prediction of the outcomes. And so these are the top 6 predictors that help the most with prediction. So for VTE, factor V Leiden, the use of topoisomerase inhibitors, apixaban use had a negative relationship, which is evident why. Stomach or pancreatic cancer, hemoglobin, and then HSCT. So these were the top predictors for VTE. And then for clinically relevant bleeding, microparticles, which is kind of written as extracellular vesicles here, NT-proBNP, CRP, age, BMI, and then platelet and white blood counts were the most important predictors here. And so to conclude this theme, with the use of advanced statistical methods, we achieved higher prediction and better performance measures. We also observed that some genetic and biomarkers, which are not yet available in routine clinical practice, helped improve prediction. And while the performance measures are encouraging, they are internally validated performance measures, and therefore external validation is needed. And so that concludes the 4 themes that were researched during my PhD. And so now, what are the clinical implications and future directions based on this research? Well, the clinical implications are that, of course, the non-O blood type, factor V Leiden, and the prothrombin gene mutations, they were important genetic risk factors during the studies, and therefore they are important genetic risk factors to potentially consider when deciding on thromboprophylaxis use. We have also identified several biomarkers which may be potential predictors, which upon confirmation could support more personalized risk stratification and guide thromboprophylaxis decisions. Then we observed that the predictive value of biomarkers may change at different time points because we did observe during the systematic review and meta-analysis that some biomarkers were predictive at the time of cancer diagnosis, others were predictive pre-chemotherapy, and others were predictive preoperatively. Then the significant associations we observed between inflammatory and cardiac biomarkers may help improve current risk prediction for VT and bleeding. And these findings suggest that perhaps we should adopt a broader perspective when assessing VT and bleeding risk in cancer patients. And then lastly, we observed that machine learning models might or may improve prediction of these complex outcomes and should maybe be used more in this setting. And so looking ahead, there are several important areas for future research. So future research could look at the advantages versus harm of inherited thrombophilia screening, can look at the cost effectiveness of inherited thrombophilia screening and biomarker testing, look more into the biological mechanisms between the associations we observed among the inflammatory and cardiac biomarkers. They could try to validate our observed associations or even the prediction models we developed. And another area of research would maybe be to look into dynamic risk assessment models as well. And so, yeah, so I'd like to acknowledge my supervisors, which have been key collaborators on my research. And I also want to acknowledge all the collaborators that have helped me throughout, and my thesis advisory committee, which have helped make this research happen. And these are a couple pictures throughout my PhD, which kind of sum up my journey of the people I've met, the conferences I was able to be a part of, and the late nights that have gotten me here. So thank you so much for listening. I'm happy to answer any questions, or if you think of questions later, feel free to contact me. I've included my email on the slide for this reason. And thank you again for the invitation to present at the seminar.
Thank you very much, Dr. Roy, for that really great presentation, and congratulations on all of these very impressive studies that you were able to churn out during your PhD. Let me just see here. There's some comments in the chat, and maybe while we're waiting for a question or two from viewers, I'll ask a few questions. So I was very interested with just the general idea, as you say, you had done maybe a scoping review of the literature, an overview, and identified genetic risk factors as an area that was under-investigated. So I was really interested to see that Factor V Leiden prothrombin gene mutation so effectively increased risk stratification, even in this patient population that's already at very high risk.
Mm-hmm.
And not only that, it seemed to also effectively translate into an increased therapeutic efficacy, right? You're seeing more bang for your buck. Outside of cancer, there's still a lot of controversy about genetic thrombophilia testing, and I know at the end you had a line about investigating like the cost effectiveness and the psychological harm. So I'm just curious whether this controversy around genetic thrombophilia testing came up with reviewers, for example, and were you surprised at the results, and how did you justify, I guess, looking into this in cancer patients?
Yeah, it did come up by a reviewer. I did have a reviewer kind of mention that including more of a discussion on screening in the article itself would be helpful. So I've included it a bit in there. Now I know the controversy is more so in the general population, right? And whether a positive inherited thrombophilia results will change management is probably less likely in the general population, right? But I think when it comes to in patients who already have a higher baseline risk, then I don't know that, I guess I'm thinking that perhaps a positive inherited thrombophilia in patients who already have a higher baseline risk might be more likely to change management compared to a patient population which have a lower baseline risk. I guess that's my opinion based on just statistics. And I guess how I kind of envision VT events is kind of using causal pies when I'm going back to my epidemiology background. And I just try to think that if the more risk factors are present within the pie itself, the more likely a VT event is to occur. So I think if the patient already has a cancer diagnosis, adding another risk factor on top of that, I would think might be more likely to make the clinician or physician change management from there. So yeah.
Yeah, thank you very much. And maybe we have one question in the Q&A, but before that, just a follow-up question. I mean, when I saw your data, my mind went to a parallel with estrogen and birth control, where there's a very well-established, maybe even a synergistic interaction if patients have Fact 5 Leiden and they're on the birth control pill. I know you weren't able to look at that with
Yeah.
AVERT because they all have cancer,
Yeah.
but just curious if you guys thought about trying to formally investigate a statistical interaction between cancer and genetic
Yeah.
thrombophilia.
So when I was doing my systematic review and meta-analysis on inherited thrombophilias, I did in fact come across studies which did look at this. I believe one is by Gran et al. Published in the year anywhere between 2015 and 2020. I can't remember the exact year. And there's another one by Skill, which I think Skill et al. I think that one's 2015 or 2020. And they looked at the synergistic interaction between a cancer diagnosis and factor V Leiden. And if I remember correctly, they report a synergistic interaction, which supports it. Um, Now, I don't remember any studies looking at prothrombin gene mutations, so that's perhaps maybe something to look into. But the tricky thing is prothrombin gene mutation is less common, so you might need quite a large sample size to assess this with enough power. But the factor V Leiden one is something that has been looked into. I don't know how big their sample size is, but the fact that there's two studies which found the same conclusion is a little bit more reassuring, right?
Hey, thank you very much. So there's one question here from Dr. Brandow. So he says, thank you for your presentation. Congratulations. Can you please expand on the topic of dynamic risk assessment models and their current stage in predicting thrombosis and bleeding outcomes?
Yeah, so this is in fact something that I'm aiming to look into more during my postdoctoral studies once I secure funding. But essentially, over the course of a cancer diagnosis, there are a lot of time varying exposures that might occur. So, for instance, a patient might get diagnosed with not an advanced cancer. They might undergo certain therapies, and then later down the line, their cancer progresses, they get surgery, and so on, in which their baseline risk then changes because they're now exposed to other VT risk factors during their journey. And this is not taken into consideration using more static baseline risk models, right? So that's kind of the area that I'm trying to go into during my postdoctoral studies, is trying to use methods which is able to not only estimate a baseline risk, but also take into consideration time varying exposures that occur throughout the cancer diagnosis and updating the baseline risks based on the development of new risk factors.
Yeah, very interesting. And I know, biomarkers, the idea of biomarkers has been around for a long time. And 10, 15 years ago, they they were helpful to some extent, but I also
Mhm.
think that there was some degree of scientific disappointment with maybe isolated measurements. But I do find it very interesting that if we're maybe better able to model what's actually happening in the real world, as you say, people change, right? And if you're
Yeah.
only measuring the information at one time point, it's never going to be perfect. Um, so maybe just one follow-up question about machine learning. We hear about AI and whether related to science or outside of science, generally now with ChatGPT, for example. And I find a lot of the time AI methods are described as a perfect solution and it's going to solve all of our problems. But one of the common downsides you mentioned, you sacrifice interpretability with these more advanced models, right? And that's why you need explainable AI. But I've often read as well that they create such a good fit to the data. It's almost like you're creating a perfect mold to the data that when you then try and validate your findings in an external cohort, that C statistic, the AUC value, then falls. So I'm wondering, again, did this come up at all in your work? And do you think that maybe we'll get better at that over time with model development? What are your thoughts on that?
Yeah. Overfitting is definitely an issue. I mean, it's also an issue with more simple models as well, right? If you add too many variables, you can overfit the data. But in both cases, with complex models such as machine learning or in simple models, there are ways to prevent it from happening. So I think like specifically in machine learning models, you can use sort of different cross-validation in order to select your hyperparameters, which are kind of like the settings to your model. You can use it to select the variables that you're including. There are also specific, well, for the XGBoost anyways, there are specific hyperparameters which you could utilize in order to prevent it from happening as well, which are lambda, alpha, and gamma, which are probably the three more common ones. And so you can tweak them. So I think as long as you're aware that it can happen and you include strategies to try and mitigate it from happening during your model pipeline, I think that's crucial to try and avoid it from happening. I mean, I know it's exciting to see a really high C statistic happen during model development. But I think, as long as you're mindful during the development of your model that overfitting can happen and you implement strategies to prevent it from happening, then that's a way to try and get machine learning models that are more externally that are externally validated with a high enough performance measure.
Okay, perfect. Well, I think at that, there's no other questions, but maybe we'll end there. I want to say thank you again, Dr. Roy, for that really fantastic presentation and discussion. And also just to say that our next seminar is going to be on February 11th. And this is going to be Dr. Abdulrahman presenting on the peri-procedural anticoagulation, the management of peri-procedural anticoagulation for central line insertion in persons with cancer. So again, thank you, Dr. Roy, for the great presentation.
Not a problem. Thank you so much.